Hello, everyone. Thank you for attending today's webinar, Human Gut Microbiome-Based Contract Research Applications in Toxicology and Microbiome-Based Drug Discovery, presented by Frontage Laboratories. I'm Ricardo Castaño, and I'll be moderating this webinar. Our speaker today is David Kwok, President and Founder of Frontage Laboratories. You can read his full bio on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. This webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. All right, now let's begin. David, please go ahead.
Okay, great. Thank you, Ricardo. Just a housekeeping item also. Ricardo introduced me as the president and founder. It should be BRI, which was a privately held contract research company, and BRI has become part of Frontage Laboratories as of 2019. So just for the record, and it's a pleasure to meet everyone online, and thank you for dialing in. I'm glad to share with all of you in this webinar, first to start off with a literature review of gut microbiome metabolism, and then applications in metabolism and gut microbiome-based drug discovery and development. In this outline, we will begin with a review of the background in gut microbiome. Then from a drug metabolism point of view, we will discuss pre-systemic contributions to metabolic clearance. Next, we will review metabolites from the gut microbiome in modulating metabolic diseases, such as type 2 diabetes. We'll take that as an example. Then we will conclude with a summary of drug microbiome metabolites and known intestinal biomarkers as tools in microbiome-based drug development. And lastly, I would be ventured in proposing drug microbiome translational strategies in enhancing clinical outcome. So next, let's begin to take a look at what we know about the gut microbiome. Now, over the past decade, or even slightly longer, a number of excellent reviews have published discussing the gut microbiome, which is considered a so-called microbiome-metabolic organ. It's actually an organ within our body. On the left-hand panel, a review of the gut microbiome ecosystem starts at the upper left hexagon, and it talks about the beginning of the gut microbiome from delivery and childbirth, and continues to evolve clockwise with our diet, our geographic locations, to the host genetics within our body, and the use of antibiotics during our lives, and then the use of prebiotics or probiotics in our diet, and then eventually our age and lifestyles all impact as variables interplay in determining our gut microbiome. So it's a very heterogeneous situation between individuals, certainly not a static environment, not a static ecosystem. Now, the panel on the right is an excellent review of the number of organisms in a healthy gut microbiome. And using modern metagenomic sequencing data, the authors in this case reported a list of 157 organisms, which then was classified into phyla, classes, orders, families, and genera and species, which when we are considering only the genomic signature. However, if we also consider not just the identity of the organisms, but also the functions by looking at their transcriptome, the mRNA data in these bacteria, in other words, looking at the diversity in enzyme functions as well, the authors reported, close to 1,000 organisms in a healthy gut microbiome. So it's again It's very complex, not just the number of organisms, but what they do as well. The majority of the organisms shown in the picture in the phylum Bacteroidetes in the blue color, followed by the phylum Firmicutes in the green. So these are the two major phyla of bacteria in our healthy gut microbiome. And this bacterial diversity is really made up by a population of over 100 trillion bacteria, containing a catalog of over 3 million bacterial genes identified back in 2010. And then in 2014, the catalog of bacterial genes was updated to over 9 million genes. Now, compared to only 22,000 genes in our human genome, this puts into perspective the complex gut microbiome for hopefully the rest of our discussion. Now, it is obviously important to know the diversity, as we talked about, and the relative abundance of bacteria at the species level in the gut microbiome, but it is equally important to discuss the functions. So here we will discuss the gut microbiome in contributing to pre-systemic drug clearance. Let's talk about gut microbiome in contributing to pre-systemic drug clearance. Now, if we follow the entire length of the gastrointestinal tract, pre-systemic metabolism of nutrients begins in the saliva containing digestive enzymes, and it continues throughout the digestive tract. Now, along the digestive tract, from the duodenum, jejunum, ileum, colon, the number of organisms, the bacteria, are increasing dramatically along the length of the GI tract. And many examples of drugs involving a large number of bacteria and bacterial enzymes have been characterized using metagenomic data and transcriptome data. Now, at Frontage Laboratories, we routinely perform in vitro gut microbiome metabolism assays to provide drug clearance data and metabolite information. Now, next, if we look at the drug, we continue with the drug absorption process. If a drug is absorbed by transcellular process, meaning it is absorbed inside the intestinal cell, then potential pre-systemic metabolism within the cells is possible. Alternatively, if drugs are absorbed passively through a so-called paracellular process, meaning that it is absorbed through the spaces between the cells, then pre-systemic metabolism may be avoided. So it really depends on the process of what's driving drug absorption through the intestine. Within the intestinal cells in the GI tract, a full complement of oxidative P450 metabolism enzymes and conjugation transferases are operating, and this leads to pre-systemic metabolic clearance. And so interestingly, an example here is the discovery of a class of polyphenols called phenylcoumarines in grapefruit juice, and that was identified in inhibiting pre-systemic intestinal metabolism of an antiplatelet aggregation drug clopidogrel or Plavix within the intestinal cells. So for us to study intestinal metabolism, now human intestinal cells or enterocytes are commercially available as cryopreserved intact cells, similar to the use of hepatocytes in supporting in vitro metabolism within the human intestine. One additional pathway for pre-systemic drug metabolism that we should talk about is when the drug is absorbed through the GI tract, draining by the mesenteric circulation into the portal vein and then to the liver. Significant metabolism through first pass of blood through the liver can also occur for some drugs before reaching systemic circulation. And this is referred as hepatic first pass metabolism. And so the drugs listed here are some of the examples of drugs exhibiting hepatic first pass metabolism. Now, in vivo models are available now to tease out the contributions of hepatic first-pass metabolism relative to systemic clearance, and this information is useful for predating the need for dose adjustments in patients with compromised liver functions. Now, today we will obviously discuss pre-systemic metabolism by the gut microbiome as our focus. Now, when we discuss drug metabolites, we should also mention a recent release of the U.S. FDA guidance on safety testings of drug metabolites, so-called the MIST guidance. This recent guidance was published in March 2020. This guidance was first drafted back in 2005 and was as a draft and finalized in 2008. And this most recent publication is aligned with the ICH guidance for non-clinical safety testing, the M3R2 guidance from ICH. Some of the important questions discussed in terms of metabolites in this guidance include the question whether there is formation of active metabolites, also whether there is enterohepatic recycling of the parent drug or the conjugate, either glucuronide conjugate or sulfate conjugate. Enterohepatic recycling is a molecularly driven process in its mechanism, and in terms of toxicity, some conjugated metabolites such as the azyl glucuronide can be reactive in nature and may warrant safety evaluations compared to other types of glucuronides, such as N-glucuronides or ether or carbyl ester glucuronides, which are typically less reactive. Another is the metabolite relative abundance data observed during general toxicology testing in animal species, such as rats or dogs. And the question whether there's human unique metabolites not present in the safety species is always a question we try to address. Or metabolites in safety species with disproportionate systemic exposure compared to that in humans. So in terms of significant metabolites with potential safety concerns, a guidance of greater than 10% systemic exposure relative to that of the parent drug at steady state is really what we use as a general cutoff, which warrants further attention and investigations. So for our discussion today, it is useful to be aware of the origin of the metabolites, whether it comes from pre-systemic pathways or systemic exposure, resulting in systemic exposure. And, starting from the gut microbiome or from intestinal cells or from hepatic first pass. So these are the major pathway of pre-systemic metabolite formation. Now, in terms of research tools that allows us to look at each one of these processes of pre-systemic drug clearance is to start off with in vitro assays, of course. In terms of in vitro assays, a study typically involves collection of the stool from healthy or patient donors. Obviously, the collection needs to observe exclusion and inclusion criteria that are developed relevant to the study. Donors may also involve tox species as well, like either the rats or the dogs, for correlation to in vivo metabolite formation from your toxicology studies. So each stool sample is collected ex vivo using an anaerobic buffer. We then follow a polling protocol carried out under anaerobic conditions to provide a fecal homogenate with known parameters such as pH enrichment or dilution based on wet weight. And this wet weight dilution is important as a surrogate label for enzyme concentration, which allows us to design relevant substrate concentrations to be used for each chemical class of test compound in relation to the enzyme pathway that is involved. And we will, take time to discuss a couple of examples in a moment. For each chemical class, then, and the known enzyme pathway in drug metabolism, we typically characterize and standardize the microbiome reaction kinetics using a probe substrate, so that we can reference that specific probe substrate in terms of enzymatic reaction rates. Then drug clearance data and metabolite ID information is generated by chromatography and mass spectrometry assays. If we are interested in knowing what are the bacteria, obviously we can use the shotgun genomic sequence assays to provide bacterial diversity and relative abundance information in the stool sample. But we do not perform this assay in-house, but arrangements are available for collaborating contract research companies to provide genomic sequencing data at the species and strain level resolution. So next we can maybe now jump into the applications of in vitro gut microbiome metabolism assays. First is the published metabolism of a complex glycoside through multiple metabolic steps, resulting in a final end metabolite. And then the second application is a well-known microbiome metabolism of an aromatic azo compound, sulfasalazine, which leads to the formation of active metabolites, the 5-amino salicylic acid and sulfapyridine, and sulfasalazine is used for the treatment of inflammatory conditions of the colon. Now, in this first example, which involves microbiome metabolism as a primary source of metabolite formation produced by intestinal bacterial enzymes, the metabolites form is then absorbed, resulting in systemic exposure. And this example is stereoglycosides, which are a group of compounds that are isolated from a plant, Robbidiana bertoni. And stereoglycosides are currently available as high potency sweeteners, about 200 to 300 times sweeter than table sugar. Now, since stereoglycosides are not at all absorbed orally, it is completely metabolized within the gut microbiome by bacterial alpha and beta glycoside hydrolases to form stevia as an end metabolite, shown in the right panel here. And stevia as a metabolite is orally absorbed, resulting in systemic exposure and susceptible to secondary metabolism in vivo, and the conjugated stevia metabolites are excreted in the urine. So we now work supporting the development of stereoglycoside as a food ingredient and a food additive. In vitro microbiome metabolism data provided the necessary stevia metabolite formation time course and mass balance information shown on the left panel here. The light blue colors are showing the relative disappearance of rebodioside A, which is a major stereoglycoside component in the extract from the plant, on the y-axis plotted against time. And the darker blue bars are data corresponding to the relative formation of a stevia metabolite over time. As you can see that the data show 100% recovery of stevia metabolite over time within a relatively short period of time. Next, as a second example in gut microbiome metabolism, it involves the metabolism of sulfasalazine, which was developed back over 50 years ago for the treatment initially for rheumatoid arthritis. Today, sulfasalazine is still currently used in the standard care for ulcerative colitis and other inflammatory conditions of the bowel. And there are now newer drugs in the same chemical class as azo compounds that are available on the market now for treating colitis. The mechanism of actions of sulfasalazine for treating colitis is not fully worked out, but it is believed to be mediated by the two major metabolites produced by the gut microbiome, 5-amino salicylic acid and sulfapyridine. Both of these metabolites are formed within the intestinal tract by bacterial azoreductases. So in our work, we utilize sulfasalazine as a probe substrate to characterize the Michaelis-Menten enzyme kinetics parameters, the KM, Vmax, to standardize the human fecal homogeneity assays involving the gut microbiome reductase activity. So in this assay, as an example, it allows us to evaluate new drug candidates as substrates of bacterial azoreductases in terms of the relative rate of active metabolite formation in the colon. And so, to recap the examples that we just talked about, it is useful to consider in vitro gut microbiome assay as a tool to provide relevant human pre-systemic clearance and colonic metabolite information prior to a first-in-human study, because this is the first piece of data we can generate that's relevant to human using human material. Second, for investigations into gut microbiome contributions in pre-systemic clearance, a relatively high enzyme concentrations along with relevant design of in vitro anaerobic conditions should be considered. And lastly, for in vitro gut microbiome metabolic stability assays, experimental design should always observe Michaelis-Menten enzyme kinetic parameters, and that is for each substrate chemical class and enzyme pathway that is defined with a fecal homogeneity concentration. So maybe for the moment, we should take a quick few seconds to look at a polling question. Great. Thank you, everyone. Now, following our earlier discussions on in vitro gut microbiome studies, I would like to discuss an in vivo model using germ-free mice and autobiotic mice to provide mechanistic evidence of a three-way interaction between dietary fibers, as an example, and butyrate or butyric acid as a microbiome metabolite, and then eventually the benefits in a disease condition, in this case, colorectal cancer efficacy. Now, germ-free mice are currently commercially available from a number of vendors, and the husbandry practices and animal use protocols are really well established. Autobiotic mice are really germ-free mice inoculated with a specific species or strain of bacteria. So by using germ-free mice in combination with autobiotic mice, we can design studies to tease out the interactions between the microbiome bacteria, the metabolites produced, and a potential therapeutic benefit. And a number of clinical studies have been investigated and reported the beneficial association between dietary fibers in colorectal cancer patients. However, in an observational clinical study, Causality between dietary fibers and cancer treatment benefits is often difficult to establish without mechanistic understanding of a specific gut microbiome metabolite from fibers in our diet. So let's review this example, what we know about dietary fibers. And first, dietary fibers are known to decrease the transit time in the colon or promote bowel movement. Second, we know that fermentation of soluble fibers in our diet also give rise to short-chain fatty acids or as metabolites. Particularly, one of the short-chain fatty acid metabolite is butyrate or butyric acid. It is beneficial for the physiology of the colon because butyrate is utilized as a primary source of energy for intestinal cells in the colon. In addition, butyrate that is formed in the colon also acts as a potent histone deacetylase inhibitor, or we call HDAC inhibitor. And a number of synthetic HDAC inhibitors have been investigated as drug candidates for treating a number of conditions, including central nervous system conditions and cancer. And the way butyrate works is, in cancer at least, is that it's absorbed within the intestinal cells in this example, and it regulates gene expression. A number of studies have explored the use of butyrate as a potential treatment for colorectal cancer over the past few years. Now, this cited study in 2014 is an in vivo study in germ-free mice and autobiotic mice inoculated with a bacterium called Butyryrhizio ribisolvens, or we call BFIB for short. This bacterium is a known butyrate producing bacterium. So by inoculating this bacterium in germ-free bowel mice, the mice were then randomized into several groups and then fed either a high fiber diet or low fiber diet. And then colorectal cancer was chemically induced in these mice using a chemical called azoxymethane in combination with dextran sodium sulfate over the course of a few months. And once the model was established, then the results from this study in the left panel, in the first cohort of mice on the left, shows that butyrate-producing bacteria was not inoculated, and in the first cohort of mice was used as a negative control. We can see that from this negative control cohort of mice, the mean tumor volume were relatively large and comparable between the low fiber and the high fiber diets. Now, in the second cohort to the right, a wild type bifidobacterium, which is a butyrate producer, it was inoculated, and the mean tumor volume in the low fiber diet mice remained large, while the high fiber diet mice in this same cohort indicated a significant reduction in mean tumor volume. In the third group of mice, third cohort to the right, a mutant strain of bifidobacterium was introduced and was observed with reduced capacity to produce butyrate, but the mean tumor volume were similarly affected by either a high fiber diet or low fiber diet. And the last cohort without bacterial inoculation, these cohort received triburine in the diet. Triburine is a naturally occurring butyrate conjugated to triglyceride found in dairy butter, and it provided a rich source of butyrate in the diet. And this last cohort was used to demonstrate that tumor volume reduction is not dependent on the bacteria itself, but it's dependent on butyrate when an alternate source of butyrate was provided in the diet. So this study also evaluated histopathology of the tumor shown in the right panel, um, where high-grade colorectal tumors, which is shown as the white portion of the bars, were least observed in beefy inoculated mice receiving a high fiber diet. So this study showed that the combination of the bacterium and a high fiber diet improved the morphology of the tumor. So in summary, this study was able to mechanistically demonstrate a direct interaction between the gut microbiome, the dietary fibers, and butyrate as a metabolite, form as a short-chain fatty acid in benefiting colorectal tumor efficacy in a germ-free and autobiotic mouse model. The authors also concluded with suggestion that dietary fibers in a form of prebiotics or probiotics approach can be explored as an adjuvant in colorectal cancer treatment. So next, I'd like to summarize the use of germ-free mice and autobiotic mouse models in microbiome study as an application. Now, first, in the colon cancer mouse model we discussed, we can investigate a possible mechanistic interaction between diet microbiome in a mouse tumor model. Second, we can utilize germ-free mice versus autobiotic mice inoculated with a bacterium as a disease model to investigate treatment efficacy. Also, thirdly, we can possibly investigate the relative in vivo contribution of drug metabolites from the host versus the gut microbiome compared to conventional mice to tease out the relative contribution from the gut microbiome. And lastly, but not the least, is that we can also utilize germ-free mice to study the role of beta-glucuronidase from bacterial enzymes in the gut to look at enterohepatic recycling mechanisms. And this approach has been used for studying a chemotherapeutic agent, irinotecan, in studying the microbiome metabolites from irinotecan in mediating intestinal toxicity, for instance. So if we could bear with one quick polling question before we move on to discuss the microbiome metabolism in human health. Great, thank you. Now, in our next discussion of gut microbiome metabolites influencing therapies and chronic diseases, and a number of comprehensive literature reviews have been published, and this is a picture of some of the common chronic conditions influenced by the gut microbiome, including obesity, diabetes, cancer, CNS conditions, and a number of CNS conditions. Respiratory conditions, including asthma, cardiovascular diseases, and also lastly, metabolic conditions such as non-alcoholic fatty liver diseases, NAFT, and including NASH, which is alcoholic steatohepatitis, a severe form of NAFT, and lastly, enterohepatic recycling. Obviously, inflammatory bowel diseases and peripheral vascular diseases should be included. And this picture points to our understanding that gut microbiome metabolites not only act locally in the intestine, but systemically throughout the body, playing a role in many chronic conditions. Now, among these chronic health conditions, I think one of the most studied is obviously obesity and type 2 diabetes. And summarized here is a recent article on a review of over 4,300 peer-reviewed articles on the topic of gut microbiome and obesity published between 2000 and 2017. Now, the bar for 2017 suddenly dropped, and that was because of the cutoff date for inclusion of the review articles. It does not reflect the fact that suddenly 2017 people stopped publishing in this area. This large interest is supported by the totality of a rapid buildup of mechanistic understanding between the gut microbiome metabolites and its functions in glucomodulation in obesity and type 2 diabetes. Now, let's start with a review of one of the working model in the relationship between obesity and type 2 diabetes. Starting from the upper left corner of this picture, normal edible tissue or fat tissue is transformed by the access nutrient, which trigger infiltration of macrophages. Macrophages are immune cells that initiate an immune inflammatory response, followed by hypoxia or the lack of oxygen, and then necrosis of the tissue and resulting in the reduction of insulin sensitivity within the fat tissue. These changes eventually result in a series of biochemical events in the muscle and the liver downstream, and this leads to elevations in free fatty acids systemically and elevations in leptin, which is a starvation hormone produced by the fat cells. And this hormone acts in the brain to stimulate the vagus nerve that innervates the GI tract, that controls the GI tract and increase our appetite. These changes leads to an overall elevation of blood glucose. And persistent elevation of blood glucose with auto-oxidation of glucose creates a persistent oxidative stress environment, which leads to insulin resistance and dysfunction of the pancreatic beta cells and eventually resulting in type 2 diabetes. So with this working model, we're beginning to understand that a high percentage of obesity will eventually lead to type 2 diabetes. Now let's discuss the established understanding between the gut microbiome, our diet, and obesity and type 2 diabetes. Now, within the colon, a dynamic ecosystem exists between the bacteria, the dietary components, and obesity, and eventually type 2 diabetes. And starting from the upper left corner of this picture, a healthy diet containing high portions of fibers, such as resistant starch and plant cellulose, all of these fibers promote a healthy gut microbiome characterized by a number of organisms such as Faecalibacterium species and a few other species listed here as examples. These organisms are known fiber fermenters, and they produce short-chain fatty acids such as acetate, propionate, and butyrate. And interestingly, the largest release of short-chain fatty acids from the gut into systemic circulation has been demonstrated to occur in the distal colon, the lower portion of the large intestine, where bacterial density is the highest in the GI tract. And we also know that short-chain fatty acids form in the proximal colon, which is the upper portion of the colon, are susceptible to circulation through the portal vein into the liver. And during blood flow through the liver, butyrate and propionate are highly extracted and metabolized by the liver. And, this is first pass metabolism, whereas acetate gets, is being extracted at a lower level and therefore reaches systemic circulation in significant higher amounts. So this is one of the reasons we commonly observe that acetate is a major short-chain fatty acid in plasma formed by the gut microbiome. And one of the main functions of short-chain fatty acids locally in the colon is their role in promoting a healthy mucus layer, like the blue-green area in this picture, which protects the colon epithelium from invasion by bacteria in the colon. Short-chain fatty acids also enhance the colon epithelial tight junction, that is the intracellular spaces between the colon epithelial cells. And this tight junction regulates permeability of the gut in terms of drug absorption, in terms of invasion by bacteria, shown here as the pink-colored cells. And on the other hand, on the right-hand side of this picture, given a diet low in fiber but high in gluten, bovine milk, or animal proteins, or food in high fat content, these dietary components promote a number of bacteria associated with dysbiosis, a term that is used in the literature to describe dysfunctional gut microbiome characterized by Bacteroides species and a few other species listed here. These organisms are in turn responsible in the metabolism of amino acids in the high-fat diet or high-protein diet, and the increase in the levels of branch-chain fatty acids such as isobutyrate or 2-methylbutyric acid or isovalerate in the gut microbiome. And persistent levels of these branch-chain fatty acids, different from the short-chain fatty acids we talked about earlier, these branch-chain fatty acids eventually lead to a decrease in the mucin production, shown here on the right-hand portion of the picture, leading to increase in gut permeability and deterioration of the tight junction between cells, and then eventually invasion of colon epithelial lining by bacteria, causing inflammation of the gut. And this situation is being referred to as a leaky gut or dysbiosis. So these are the events, the way our diet alters our gut microbiome and in turn affects the colon physiology and brings on a number of chronic diseases, including obesity and diabetes. And next, to discuss the gut microbiome metabolites, we want to take a look at this complex picture. This complex picture does reflect the complex interactions between microbiome metabolites at the local level in the gut, or systemically, these metabolites act in other organ systems. Now, to begin from the left side of this picture, short-chain fatty acids that reach systemic circulation can act on adipose tissues, mediating the release of leptin, which is a satiety hormone released by adipose tissues, fat cells. And within the adipose tissues on the left bottom, short-chain fatty acids regulate energy metabolism and thermoregulation, labeled here as thermogenesis. Now, in terms of the pancreas, there's only some indications of short-chain fatty acids systemically having a direct stimulatory effect on beta cells in the pancreas in modulating insulin and release in blood glucose. Back to the top of this picture, the short-chain fatty acids produced by bacteria, namely propionate, butyrate, and succinate, all of these have been reported to be involved in gluconeogenesis, which regulates a beneficial decrease in glucose production in the liver. Now, locally in the colon, short-chain fatty acids can activate G protein-coupled receptors, so it's so-called GPCRs. Labeled here in the picture is GPR41 and GPR43. These two receptors belong to a large families of signaling proteins that distribute in many organ systems that modulate cellular response to hormones, cytokines, and neurotransmitters. And so these receptors are important targets in drug development. And short-chain fatty acids such as acetate, propionate, and butyrate, based on the arrows in this picture, can act on GPR41 and 43. And as shown in the picture, they they mediate the release of GLP-1, which is glucagon-like peptide, and also PYY, which is peptide YY, produced by the intestine and produced by specialized intestinal cells. Now, both GLP-1 and PYY are the two gut hormones that is important in blood glucose modulation, controlling insulin release, and appetite regulation. In addition to activating the GPR receptors and involving gut hormone release, GPR receptor also has a direct effect in the central nervous system. Short-chain fatty acids can distribute into the brain and modulate neurotransmitter release, GABA, serotonin, and catecholamines. And these neurotransmitters regulate our appetite and also energy metabolism in obesity. Another class of important microbiome metabolites are the primary bile acids produced in the liver. One of the important bile acids produced in the liver is folate or cholic acid on the upper right-hand corner of the picture. And cholic acid is metabolized by bacteria further into deoxycholate as a signaling active metabolite. And this bile acid metabolite formed by bacteria can activate another G protein receptor called TGR5, which is present in specialized intestinal cells. And activation of TGR5 by the bile acid produced by bacteria is known to trigger the release of intestinal GLP-1 and in turn regulates insulin release and lower blood glucose. So the bile acids actually also can activate another nuclear receptor called FXR in the liver, and activation of this nuclear receptor also played a crucial role in fat metabolism in the liver. So it's been proposed in the literature that FXR activation is an important regulatory target and therapeutic novel target for delaying the progression of liver diseases, including NAF and NASH. And currently, NASH is one of the severe form of non-alcoholic fatty liver disease. Is one of the contributing causes of liver failure, resulting in the need for patients to require liver transplant as the only treatment options. So another biomarker is bacterial lipopolysaccharide, or LPS, which is LPS is the bacterial cell wall derived from gram-negative organisms. And LPS is known to stimulate so-called toll-like receptors, or TLR4 specifically, which is an important regulator of intestinal inflammatory cytokine production, resulting in inflammation of the colon. And it affects the permeability of the colon, the tight junction, and as well as invasion of the colon epithelia by bacteria. Another metabolite on the upper left corner that is important to talk about is as it relates to cardiovascular diseases is TMA, or trimethylamine. The gut microbiome metabolize red meats, and specifically choline as a component of phospholipids in meats, giving rise to the metabolite trimethylamine, or TMA. Now, TMA produced by bacteria in the intestine is readily absorbed from the GI tract, and then it's further metabolized to trimethylamine oxide, or TMAO, which is linked to an increased risk in cardiovascular diseases as a long-term comorbidity factor in type 2 diabetes. And lastly, in terms of microbiome metabolites that impact kidney functions, is also shown in this picture on the right-hand edge, is indoxysulfate and cresol sulfate, which are the two gut microbiome metabolites that recognize as toxic to the kidneys. And these toxic metabolites resulting from bacterial metabolism of proteins in our diets are particularly important for patients with compromised renal functions. So, given this complexity in the gut microbiome, here we summarize the short list of gut microbiome-based therapeutic areas and health status against the known gut microbiome metabolites and biomarkers we discussed. Here we also include a listing of the established assays by either chromatography, tandem mass spectrometry, or conventional ELISA or multiplex ELISA assays. I won't have time to go into the therapeutic areas and the metabolites assays involved, but I just want to share and provide you this information as a reference. And this information, this short list, is not meant to be an exhaustive list of assays available. And next, I'd like to share with you also a report. And as an example on metformin, which is widely prescribed as an orally effective medication for type 2 diabetes. And a number of authors actually have reported that daily oral metformin therapy in type 2 diabetic patients for 4 months showed that metformin had a strong effect on the gut microbiome. And fecal transplantation from metformin-treated patients, donors to germ-free mice showed that glucose tolerance was transferable from the patients to mice. And so the authors concluded that metformin activity in mediating type 2 diabetes blood glucose lowering is mediated at least partially through suppressing mucin bacteria, mucin-producing bacteria, and increasing the production of propionate and butyrate short-chain fatty acids. And actually, we will talk about, and that's my last slides here, that I'd like to conclude with a suggestion in terms of drug development strategies that can incorporate the gut microbiome. So we can take specifically a molecular target, for example, associated with type 2 diabetes or NAFT. Then we can envision strategies incorporating the gut microbiome through the use of prebiotics or probiotics, and then understanding the microbial diversity and abundance in the patient populations that we're studying by evaluating microbiome metabolites, and then personalized microbiome-based drug therapies. And as I'm running short on time, I would stop here. And so I thank you all for dialing in to this discussion. And if you mind to bear with me to take one last poll questions, and then we can open up for questions and answers. Great. So we are open for questions, Dan.
All right, everybody, thank you so much. Let's move on to the Q&A. There is still time to submit your questions using the questions and answers tab on the left side of your screen. We have a lot of great questions today, and we will try to get to as many as possible. And let's see, our first question here asks, David, you mentioned about first-pass metabolism by the liver. Do you have an animal model to evaluate if first-pass metabolism is occurring? Or how about an animal model to determine metabolite contribution for microbiome metabolism versus host metabolism?
Right. Yes, thank you for the questions. And yes, so rodent models are available, and typically rat are available to with surgical interventions to tease out the contributions of intestinal the first pass or hepatic first pass metabolism. We talked about microbiome metabolism, which is upstream to intestinal first pass metabolism, right? So in a rat model, we can double, like what's described in the literature is that we, double cannulated rat model is available both in the portal vein and in the duodenum. And using these model and proper study designed with different route of administrations, we can then collect serial blood samples and then assay drug exposure resulting from different route of administration to determine the relative contributions that way. In terms of the second part, in metabolite contributions from microbiome metabolism versus host metabolism, either through the liver or other parts of the body, we can briefly touch on, I think we did in the presentation talk on the use of germ-free mice to provide systemic exposure in the absence of microbiome metabolism. And then we obviously can compare it to the exposure resulting from conventional mice. In both cases, oral administration and again collect blood samples and measure exposure, systemic exposure, and beginning to tease out the contribution from gut microbiome metabolism.
All right, thank you very much. Let's see, our next question asks, from the gut microbiome metabolite and biomarkers assays available at Frontage, are these validated assays?
Well, it's a good question, and often when we first started with a lot of project, it's very early on in the stage of research, and as a research assay performance will need to be pre-validated or qualified as a fit-for-purpose assay. And that is based on a specific sample matrix, either a stool or a plasma sample. However, as a pivotal metabolite assay or a biomarker assay associated with clinical outcome or associated with human safety interpretation, then a fully validated assay is required, and we would obviously advise the sponsor to perform a validation if it's appropriate.
All right, thank you. Now let's see. This will probably end up being our last question here. We do thank you for all the great questions that we did have. We couldn't get to them all, but we will try our best to get back to everyone who submitted personally after the webinar. Now let's see. This question asks, where can you measure SCFAs and BCFAs as biomarkers in the blood? Is it in the gut lumen or the gut mucosa?
Yes, we can actually measure both short-chain fatty acids and branched-chain fatty acids in the blood as a systemic exposure readout of these types of metabolites derived from the gut microbiome, as well as the gut lumen. And obviously, the data that we derive from the gut lumen, meaning from stool samples, tends to be a bit more variable, right? And one thing about these types of assays is that there's always a circulating background level, endogenous level, and so it depends on the study design. If there's a specific treatment interventions that we are interested in studying, we need to keep that in mind, like what are the endogenous circulating levels either in plasma or in the gut lumen in the stool sample, and whether those interventions would give rise to significant changes that we can be able to tell very confidently resulting from the interventions that we're trying to study. In terms of the mucosa, it is more difficult unless we have surgical models that we actually go into necropsy of the animals and collect the mucosa and then look at the formation at the mucosa level mechanistically. But most commonly, we just look at either the stool samples or the blood samples.
All right, thank you so much, David, and thank you all for attending this Fierce Bio for submitting so many great questions. I'd like to thank David for participating again, and I'd like to thank Frontage Laboratories for presenting today's webinar. And remember, this webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.